Hello friends, welcome to Biohacks. I am Solanki and today we are going to discuss about blood groups. So before going to the discussion, let us know why blood groups are so important. Blood groups are important because during blood transfusion, if the blood groups are not matched, then it could be fatal. Now, there are two types of blood grouping systems, ABO and RH. First of all, we will look into the ABO group. Now, have you ever thought that why people have different types of blood groups? This is because this ABO system depends upon two things. Number one, the antigens present on RBC, and number two, the antibody in the plasma. Okay, now come to this chart. See, according to the ABO system, the blood groups are of four types: A, B, AB and O. Now, let us look into the antigens present. Number 1 point. The antigens present on the surface of RBC, if it is A, it will be of A blood group. Exactly the same. If it is B, then it will be B blood group. If the antigen present on RBC is both A and B, then it is AB blood group. And if there are no antigens on the surface of RBC, it will be O blood group. Okay. Now let us come to the antibodies. This is very important that the antibodies are exactly the opposite of the antigens present. See, if it is A blood group, A antigen is present and it is anti-B antibody. If it is B blood group, B antigen and it is anti-A antibody. If it is AB, A and B both antigens present, so there are no antibodies in the plasma. And if it is O, there are both anti-A and anti-B antibodies in the plasma. Now, let me clear some doubts here. There are two doubts. Number one, where does these antigens come from and how do they determine the blood groups? This blood grouping by the antigen is genetically determined. Genetically determined. Okay, that is from birth we have all the antigens AB present in our system, and so they genetically determine the blood groups AB and O. Now, second doubt here is where does these antibodies come from? Okay. And why it is anti-B, anti-A like this. Let us see this. When we take birth, after our birth, we are exposed to the environment. Okay? Environment. And in this environment, there are both A antigen and B antigens present. Both are present in the environment. Now, here one point is very important, that our body induces antibodies in response to the foreign antigens. Okay, only in response to the foreign antigens. Now, let us suppose there is a person with blood group A. When A antigen enters A blood group, it is not foreign to it because the A antigen is already present within it before. Now, so there will be no reaction, no antigen antibody reaction or antibody production. Now, if B antigen enters, what happens? B antigen is foreign to the body of a person who is of blood group A, because B antigen is not present within. So, there will be anti-B antibody production anti-B antibody production against the B antigen. So, is it clear that why A blood group have anti-B antibodies? And similarly, B have anti-A antibodies, AB does not have, and O both anti-A and anti-B antibodies. Okay. Now, there are two words, universal donor and universal acceptor. Universal donor and universal acceptor. What are these? Blood group O is called universal donor. Why? Because it can donate its blood to any of the four blood groups. A, B, AB and O can accept blood from blood group O. And AB blood group is called universal acceptor. Why? Because AB can accept any blood group, both A, B, AB and O. All four blood groups it can accept. So now come here to the donor group. A can accept both A and O. B can accept from both B and O. AB can accept all the four blood groups because it is universal acceptor, and O can only accept from O. Is it clear? Now, here one more doubt can arise, that why O blood group can donate to each and every blood group in spite of having anti-A and anti-B antibodies within it. Why is this so? Let us know this. Now, suppose there is a recipient and a donor. The donor is of course O and the recipient, let it be A. This A recipient have A antigen within it and O have anti-A and anti-B antibodies. Now what happens? If the donor O gives the blood to recipient A, then here antigen antibody reaction should occur. According to this, antigen antibody reaction should occur, but this does not occur. This does not take place. Why? Because this O blood is minimal in quantity. Minimal in quantity. So what happens? This gets diluted. Diluted, and so this antigen antibody reaction does not take place. Okay. Now let us come to the Rh group. This Rh antigen comes from the rhesus monkey. Okay, it is present in almost 80% of humans. It is responsible for positive and negative blood groups, and this depending upon what? Depending upon the presence or absence of an Rh antigen on the surface of RBC. Now, because of this Rh group is also matched during blood transfusion. Okay. Now, here we will see a special case of Rh incompatibility. This is very, very important for the exams. Okay. So, let us look into it. If suppose a pregnant mother who is Rh negative carries a fetus who is Rh positive. Now, during the first baby, what happens during pregnancy and during delivery? We will see it separately. First of all, during pregnancy, the mother and the fetus is separated by the placenta. So, what happens? The mother does not carry the Rh antigen, Rh negative, fetus is Rh positive, carries the Rh antigen, but because it is separated by placenta, the Rh antigen from the fetus does not pass into the mother. Rh antigen does not pass. Okay. Now, what happens during delivery? During delivery, there is a placental rupture. The placenta ruptures, and so what happens? Some of the fetal antigen, Rh antigen, passes to mother. Some of the fetal Rh antigen passes to mother. Now, these Rh antigens in mother induces Rh antibody production. Rh antibody production. Okay. Now, the baby is separated immediately from the mother, and so the baby is safe. First baby is safe. Okay. Now, if the mother who is Rh negative is again pregnant with the second baby, second fetus who is Rh positive. Rh negative mother does not carry Rh antigen, but she is carrying the Rh antibodies from the previous case. Rh antibodies present in mother. Okay. And the fetus is Rh positive, carrying the Rh antigen. Now what happens? Both are separated by placenta, but in this case what happens? The Rh antibodies present in mother passes to the fetus. Okay. Why? Because the antibody is smaller in diameter. So what happens here? The placenta, though they are separated by placenta, the Rh antibody passes to the fetus because of its small diameter. Okay. No, so what happens next? RBC destruction occurs. RBC is destroyed, and this causes anemia and jaundice in the baby. Okay. This disease is called erythroblastosis. Blastosis fetalis. Erythroblastosis fetalis. Okay. Now, how can be this prevented? This can be prevented if we inject the mother after the first delivery. We inject the mother immediately with anti-Rh antibodies. If we immediately inject the mother with anti-Rh antibodies after the first delivery, then the second baby can also be saved. Okay. Thank you, and please, if you like this channel, subscribe it and share it with your friends.